Look at this ball of cells. It is an early embryo, a blastula, and to a naked eye, every cell in it looks more or less identical. But here's the astonishing part, their futures are already sorted out. This patch of cells is destined to become the brain. That patch will become the gut. Another will become a muscle. Another, the skin. The cells don't look different yet, but their fates are effectively assigned. So, how could an embryologist 100 years ago, with no DNA sequencing, no fluorescent markers, possibly know which cell was going to become what? They couldn't cut the embryo open and look. That would kill it. They need a way to label a cell, let the embryo grow, and then find where that label ended up. That method gave us one of the fundamental tools of the developmental biology, the fate map. In this video, we'll define exactly what a fate map is, why it matters, and then work through every method used to construct one. From natural pigments to vital dyes to carbon particles to the genetic markers. Let's begin. Let's start with the definition. A fate map is a diagrammatical representation of the prospective fate of each part of an embryo at an early stage. So a fate map is essentially a label diagram drawn onto the early embryo, the egg, the blastula or the early gastrula that colors in and names each region according to what it will eventually become. Two words are doing heavy lifting here and you must understand them. Perspective or presumptive, meaning expected or destined. A region's presumptive fate is what it will normally become if the embryo is left to develop undisturbed. The presumptive notochord, the presumptive neural plate, and so on. Now, why do we bother? Fate maps are essential tool because they provide information on which position of the embryo will normally become which larval or adult structure. In other words, they connect a location in the early embryo to a destination in the finished animal. They tell you if you want to find the future heart, look here. Fate map is a method used in developmental biology to study the embryonic origin of various adult tissues and structures. A crucial distinction, fate versus potency. A fate map records what a region normally does. It's fate. But this is not the same as everything it is capable of doing. It's potency or potential. A cell's fate is the road it normally travels. Its potency is every road it could travel if you move it somewhere else. Early embryonic cells are often capable of far more than their fate suggests, which is exactly why if you transplant a presumptive skin cell into a presumptive brain region early enough, it may happily become a brain instead. Fate mapping of different types of animal can be constructed by the following methods. The central challenge in all of them is the same. Three conditions. It must stay attached to the cell and its descendants. Second, it must not harm the cell or interfere with the normal development. Third, it must be visible or detachable later, so you can trace where those cells went. Naturally sometimes provides the marker for free. When it doesn't, we supply one artificially. So the method split into two broader families, natural marking and artificial marking. In natural marking, we are using the embryo's own pigments, but in artificial marking, we are using vital dyes, carbon particles, and genetic markers. So let's take them in turn. First method, construction of fate mapping by natural marking or observing living embryo. The first method needs no intervention at all. It relies on the fact that some embryos came pre-labeled by nature. The principle is different cells contain different pigments and they form different organ-specific areas. In other words, in certain animals, the cytoplasm destined for one organ is naturally a different color from the cytoplasm destined for another. The classic example is the muscle forming cells always have a yellow color. This comes from one of the most famous embryo in developmental biology, the egg of the ascidian. Its egg contains several naturally colored cytoplasm and most strikingly a patch of yellow cytoplasm is called yellow crescent. As development proceeds, that yellow cytoplasm is reliably inherited by the cells that forms the muscle. Yellow region in the egg is the muscle in the larva, and every single time. How the map is made? Because of natural differential pigmentation, the fate map can be constructed by observing the embryos directly, and the fate of the presumptive or prospective organ forming regions can be traced. So the embryologist simply watches, no dyes, no surgery required. They follow the colored patches with the microscope as the embryo divides and rearranges, and record where each color ends up. Yellow tracks to the muscle, grey tracks to the notochord and neural tissue, clear tracks to the other structures. This is exactly how Edwin Conklin in 1905 produced one of the first ever fate maps, tracing the ascidian embryo cell by cell purely from its natural colors. The catch here is this method is very beautiful, but it's very rare. Most embryos, including the amphibians and the birds you study most, are not conveniently color coded. Their cytoplasm is fairly uniform. So, for the vast majority of animals, nature gives us nothing to follow, and we have to add a marker ourselves. The natural marking due to pigmentation of egg cytoplasm does not occur in some animals, and in these cases, some artificial marking method has been used to construct the fate maps. There are three artificial methods. We'll take them from the classic to the modern. The first one is vital dye staining method. This is the most important method historically, the one that gave us first proper amphibian fate map. So let's learn it thoroughly. The mechanism is this method involves the use of vital dye, which stain the cells without killing them. That word vital is the whole point. It means living. A vital dye is one of that colors a living cell without killing it. Unlike ordinary histological stains, which only works on dead and fixed tissues. And critically, the dye is retained by cells for a considerable period of time without interfering with the normal process of the development. So the cell keeps living, keeps dividing, keeps moving on its natural destination, carrying the same dye with the whole way. Now you will ask how the map is built. The persistence of the dye makes it possible to establish the ultimate location in the embryo from which a fate map can be constructed. You stay in a small region, let the embryo develop, see where the color has traveled, and record it. Repeat for many regions and then you assemble it and you have a complete map. This technique is inseparable from one name, Walter Wolth. In 1920, his method was ingenious in its simplicity. He soaked a tiny chip of agar in vital dye, Nile blue sulfate, and neutral red. He pressed a dye chip against a specific spot on the surface of living amphibian gastrula, and the dye diffuses into those few cells, staining them locally. He let the embryo develop normally. He tracked where each colored patch traveled during gastrulation and recorded its final destination. By repeating this across the whole surface of the embryo, Vogt produced the first complete fate map of the amphibian embryo, and in doing so, he actually revealed the movement of gastrulation itself. Watching stained cells steam inwards over the blastopore lip, this is why the technique is sometimes called Vogt's method of the local vital staining. The limitation of vital dye is the dye gets diluted at every cell division. A fixed amount of dye split between two or more cells, so it eventually fades and cannot be followed indefinitely. Good for the medium term, not for tracing a very long lineage. The second artificial method swaps a chemical stain for a physical one. The mechanism is applying a tiny particle of carbon to the surface of the embryo. It sticks to the surface of the cell and can thus be used as a marker, enabling the investigator to follow the movement of cells and to draw a fate map. So instead of dyeing the cell, you stick a visible speck onto it. As the marker cells crawl and rearrange during development, the carbon specks rides along on the surface and you simply watch where it goes. It's especially useful for following the surface movement of the cell sheet during the gastrulation. Its key limitation is that mechanically only the fate of particular group of cells can be identified, but not the whole. Because the carbon sits on the outer surface and marks a clump of cells rather than one cell precisely, it can only tell you the fate of a group and only a surface cell. Since it can't reach cells deep inside, it is a blunted instrument than a vital die. Good for tracing sheets of tissues, but poor for resolving individual cell lineage. The third method is the most powerful and the most modern, and it solves the fatal flaw of dyes and carbon markers that fade off or wash off. The key principle is a specific DNA sequence with a known location on a chromosome which can be used in a fate mapping. This is the game changer. A dye halves with every division, but a gene is copied at every division. So every single descendant of a marker cell inherits the same marker permanently. No matter how many times it divides or how far it travels, you can trace a lineage across the entire development of the animal, not just a few days. Transplantation between species. The most famous qualic chick chimera developed by Nicole Dorin. Quail cells have a distinctly marked condensed nucleus, so if you graft a piece of quail embryo into a chick embryo, you can later identify exactly which adult tissue came from quail's graft, cell by cell under the microscope. This mapped the origin of the neural crest and much of the vertebrate body. Radioactive labeling or autoradiography. Cells are tagged with a radioactive marker such as tritiated thymidine, which gets built into their DNA and can be detected later on film. Fluorescent reporter gene. The modern standard. A gene such as GFP, green fluorescent protein, is switched on in specific cells so they and all their descendants literally glow green and can be followed in living transparent embryo in real time. This is how much of today's fate mapping in zebrafish and in mice is done. The overall trajectory is clear: watching natural colors, to painting cells with dyes, and to build the label into the genome itself. Each steps give a marker that lasts longer and resolves finer. Let's just quickly recap. A fate map is a diagram of early embryo, egg, blastula or gastrula, showing the presumptive fate of each region, what it will normally become. Remember, it records fate, not the fully potency of a cell. The methods we have seen was natural marking and artificial marking. In natural marking, we are using the embryo's own pigment. For example, the yellow muscle forming cytoplasm of the ascidian egg. In artificial marking, where nature gives no color, we have seen vital dye staining, carbon particle, and genetic marking. Vital dye staining. Dyes like Nile blue sulfate stain living cells without harming them. Volt's classic amphibian map. The limitation is dye dilutes with the division. Carbon particle marking. Carbon specks struck to the surface track cells movement, but only the fate of group of surface cells can be found. The last was genetic marking, a heritable DNA marker that is never lost, giving permanent single cell whole lifetime tracing, which is the modern standard gold. If fate maps now make sense as a single idea rather than a list of methods, that is exactly what our channel is for. So keep learning, keep achieving. I'll see you in the next one. Thank you.